Hello all of you. Today I would like to talk about the another important biomolecules, that is nucleic acids. And this is the structure of the important nucleic acid, that is DNA, and this is the double helical structure of DNA, and this is the real picture of DNA which has been captured by atomic force microscopy. So you can note the similarities between this picture and this picture of DNA. So you can also notice the double helical nature of the structure of DNA in these two pictures. So first of all, I would like to define the nucleic acid. So nucleic acid is an important class of macromolecules found in all cells and viruses. And the function of nucleic acids have to do with the storage and expression of genetic information. And the important examples of nucleic acids are DNA and RNA. DNA is the deoxyribonucleic acid, which generally encodes the information the cell needs to make protein. And nucleic acid is generally considered as the polymer of nucleotide. So the unit of nucleic acid is nucleotide. Nucleic acid was first discovered by Johann Friedrich Mischer, and he discovered nucleic acid in white blood cell, in the nucleus of white blood cell. And first of all, he called that biomolecule nuclein because it has been extracted from the nucleus of a white blood cell. So he DNA was discovered in 1869 by this scientist whose name was is Johann Friedrich Mischer, and he was a Swiss researcher. So this is the most important type of nucleic acid, that is DNA. And as you all know that DNA carries genetic information, and these genetic information generally transcribed into RNA, and RNA ultimately leads to uh, the translation. RNA generally translated a specific in a specific protein, which ultimately control a particular pathway in the biological cell, or you can say in biological system. So now I would like to talk about the importance of nucleic acid in the biological system. The first and most important function of this nucleic acid is that it helps in the storage of genetic information. As I told you earlier, that a specific segment of DNA carries the genetic information, which we generally transcribe into mRNA, which is also an important nucleic acid. And this mRNA generally translated into a specific protein, which generally helps in controlling or regulating a particular pathway or metabolic pathway in our biological system. Another important function of the nucleic acid is that it acts as the energy currency of the cell. So the most common example of energy currency of the cell is adenosine triphosphate. In short, it is also known as ATP. And in ATP, these phosphate bonds, which this phosphate group, which are joined together by phosphoanhydride bond, has generally possesses energy, and when it breaks, it releases a large amount of energy, which ultimately helps in the various anabolic processes in the biological system. Third important function of the nucleic acid, that it forms a part of coenzymes, and the most important coenzymes are NAD plus, NADP plus coenzyme A, etc. And coenzyme is an inorganic non-protein compound that binds with an enzyme to catalyze a reaction. And fourth important function of the nucleic acid that it acts as second messenger in signal transduction pathway. So second messenger are the intracellular signaling molecule released by the cell in response to exposure to extracellular signaling molecules. And it is synthesized from adenosine triphosphate, and cyclic adenosine monophosphate is involved in the regulation of glycogen, sugar, and lipid metabolism. And cyclic AMP may affect brain function in many ways. So this is all about the important functions of nucleic acid in biological system. Now I would like to talk about the unit of nucleic acid, that is nucleotides. So nucleotides are the basic building block of nucleic acid, and this is the structure of nucleotide. And nucleotides are generally consist of a nitrogenous base, a pentose sugar, and a phosphate group. So in RNA, that is ribonucleic acid, ribose sugar generally present, but in case of DNA, deoxyribose sugar generally present. So we will discuss about the difference between ribose and deoxyribose in the latter part of my presentation. So here I would like to first of all I would like to discuss about the one of the important constituent of the nucleotide, that is nitrogenous base. So it is a set of 5 nitrogenous bases, and it is used in the construction of nucleotides, which in turn build up nucleic acids like DNA and RNA. And these nitrogenous bases are adenine, uracil, guanine, thymine, and cytosine. And the basic property derives from the lone pair on the nitrogenous base. So nitrogenous bases are basic in nature, and basicity of this nitrogenous base generally depends on the lone pair of electron present in the nitrogen in the 9th position. Now I would like to talk about the types of nitrogenous base. And nitrogenous bases present in the nucleotides can be grouped into 2 categories. First is purines, and the example of purines are adenine and guanine. And second is pyrimidine, and the example of pyrimidine are cytosine, thymine, and uracil. So let's first discuss about the purines, and this is the structure of 2 purines, that is adenine and guanine. And adenine can also be named as 6-aminopurine, and guanine can also be named as 2-amino-6-oxypurines. And purines are generally cyclic in nature, and numbering of purines are generally we can number each atom in purines in anti-clockwise direction. So you can see that nitrogen, the number of nitrogen is 1, then 2, then this nitrogen is 3, and each atom in this nitrogenous base has numbered in anti-clockwise direction. Now let's discuss about pyrimidines. There are 3 kinds of pyrimidines. First is cytosine. Cytosine can also be named as 2-oxy-4-aminopyrimidines. Second is thymine. Thymine can also be named as 2,4-dioxy-5-methylpyrimidine. And third is uracil, which can also be named as 2,4-dioxypyrimidine. So these are the 3 important pyrimidines, and pyrimidines are generally homocyclic in nature, right? And they are numbered in clockwise direction. So you can see here that the number of this nitrogen is 1, then this carbon is, and the number of this carbon is set 2 and this nitrogen 3, then this carbon is 4. So the numbering of each atom in this nitrogenous base is in clockwise direction, right? So these are the way by which you can differentiate, or you can find differences between pure ions and pyrimidines. And what are those? The pure ions are heterocyclic in nature, but pyrimidines are homocyclic in nature. Pure ions can be numbered in anti-clockwise direction, but pyrimidines can be numbered in clockwise direction. Now I would like to talk about the basic uh, important properties of the nucleotides. And first and most important property of the nucleotide is that they are basic in nature, and basic property of nucleotides are due to the presence of the lone pair of electron in the nitrogen atom. Second main property of these nucleotides are that uh, the purines and pyrimidines common in DNA and RNA are aromatic and conjugated molecules. That's why nucleotides can absorb ultraviolet light. So you can see here there are few nucleotides here, and first nucleotide is AMP, and AMP is adenosine monophosphate, and here the molar extinction coefficient at 260 nanometer is maximum for adenosine monophosphate, which means that absorbance of that wavelength of light by uh, AMP is maximum here, because here absorbance of particular wavelength of light is directly proportional to the molar extinction coefficient. So that's why AMP has maximum absorbance at 260 nanometer of light, and 260 nanometer of light generally resembles the ultraviolet lights. Free purines and pyrimidine bases may exist in two or more tautomeric forms depending on pH. Uracil, for example, occurs in lactam, lactim, and double lactam form. So they exist in nitrogenous base, generally exist in two or more tautomeric forms, which generally depends on the pH. Nitrogenous bases are hydrophobic and relatively insoluble in water near neutral pH, and at acidic or alkaline pH the bases become charged and their solubility in water increases. And they are capable of forming hydrogen bonds. The functional group of present in nitrogenous base generally helps in the formation of hydrogen form. Actually, these are the properties of nitrogenous bases, not nucleotides. So in the slide you can change here, that you can write here nitrogenous base instead of nucleotides, right? So this is so name of the nucleotides here by mistake, just change it with nitrogenous base. So these are the properties of the nitrogenous base. Now I would like to talk about the ribose sugar. So the second constituent of the nucleotide is ribose sugar, and ribose sugar generally forms the backbone of nucleic acids such as DNA and RNA. And in DNA, deoxyribose generally present, that is D2-deoxyribose, and in that ribose at the second carbon, in spite of hydroxyl group, hydrogen atom generally present. So that's why these are known as deoxyribose. But in the D-ribose sugar, hydroxyl group present in the second carbon atom. So D-ribose generally present in RNA, and D2-deoxyribose generally present in DNA. And this is also the main difference between DNA and RNA. So these are the structure of nucleotides which is present in uh, RNA, and you can easily observe here that it contains ribose only, which has hydroxyl group at its second carbon atom. But in nucleotides present in DNA, you can easily observe the hydrogen group attached with the second carbon atom instead of the hydroxyl group. So that's why the ribose sugar present in the nucleotides of DNA is deoxyribose. Now I would like to talk about the phosphate group, which is an important constituent of nucleotides, and phosphate group generally attached with the fifth carbon of deoxyribose. And again, sugar phosphate backbone forms the structural framework of nucleic acid, including DNA and RNA, and this backbone is composed of alternating sugar and phosphate groups and defines directionality of the molecule. So we will discuss about how sugar phosphate define the directionality of the nucleic acid in the latter part of my presentation. So the a symbol for the nucleotide is DAMP. If adenine present as a nitrogenous base, if the guanine present as a nitrogenous base, then this is known as DGMP. Likewise, if cytosine present as a nitrogenous base, then it is known as DCMP. These are the nucleotides which are generally present in DNA, because you can easily observe here a D here. D means deoxy. Now I would like to talk about the bonds through which nitrogenous base and phosphate group attached with this ribose or deoxyribose sugar. So the nitrogenous base generally attaches with the ribose by beta N glycosidic bond, and this bond generally forms between carbon number 1 of the ribose and nitrogen number 9 of adenine group, that is purine, purines, right? And again, if in a nucleotide pyrimidine nitrogenous base present, then the N beta glycosidic bond forms between the carbon number 1 of the ribose and nitrogen number 1 of the pyrimidine. And phosphate group generally attached with this ribose sugar at carbon number 5, and the bond by which it generally attaches with the ribose or deoxyribose is known as ester bond. So this is the table which depicts the principal bases, nucleosides and nucleotides. So these are the nucleotides which generally present in the RNA, that is ribonucleic acids, and these are the nucleotides which generally present in DNA, that is deoxyribonucleic acids, and these are the abbreviation for the nucleotides present in RNA, and these are the abbreviation for the nucleotides present in the DNA. Now, there are other biologically important bases also in the biological system, and the bases such as hypoxanthine, xanthine, and uric acid are present in the free state in the cells. The former two are the intermediates in purine synthesis, while uric acid is the end product of purine degradation. So you will have to just remember the name of these nitrogenous bases. Now I would like to talk about nucleoside. Nucleoside generally consists of the nitrogenous base covalently attached to the sugar but without a phosphate group attached at the carbon number 5 of the ribose or deoxyribose. So how nucleoside and nucleotide are different from each other? They are different from each other only in respect of the presence of phosphate group, right? So nucleosides generally don't have any phosphate group at the carbon number 5 of ribose sugar, but nucleotide generally contain phosphate group at the carbon number 5 of the ribose sugar. Now I would like to talk about the different types of nucleotides in detail. So there are two types of nucleotides. Sorry, now I would like to talk about the types of nucleic acids. So there are two types of nucleic acids. First is DNA, that is deoxyribonucleic acid, and second is RNA, that is ribonucleic acid. So first of all, I would like to talk about deoxyribonucleic acid. So deoxyribonucleic acid is a polymer of deoxyribonucleotides, and it is composed of monomeric units like DAMP, DCMP, DGMP and DTMP. These are the nucleotides present in DNA, and the monomeric deoxyribonucleotides in DNA are joined together by phosphodiester bond. So this is the phosphodiester bond. The two nucleotides, or you can say two deoxy nucleotides are generally joined together by the phosphate bond, and phosphate bonds are has been formed between the carbon number 5 of the ribose of first nucleotide and present at the third carbon the ribose of the second nucleotides. So by this way phosphodiester bonds have been formed between the nucleotides in DNA. And DNA structure is often represented in a shorthand form. This is the shorthand form of the DNA, and the horizontal line generally indicates the carbon chain of sugar with base attached to carbon number 1. Near the middle of the horizontal line is carbon number 3 phosphate linkage, while at the other end of the line is carbon number 5 phosphate linkage. So this is the shorthand representation of the nucleic acids such as DNA. Now I would like to talk about the Chargaff's rule of DNA composition. And Irving Chargaff in late 1940s quantitatively analyzed the DNA hydrolysate from different species, and he observed that in all species DNA had equal number of adenine and thymine and equal number of guanine and cytosine, and this is known as Chargaff's rule of molar equivalence between purines and pyrimidines in DNA structure. So this is the question which I have asked in my class, that is, if the DNA molecule has 20% of adenine, then what will be the percentage of the cytosine? So just assume that a DNA has 100% of the nitrogenous base, and if 20% of the adenine present in that DNA, then what will be the percentage of thymine? The percentage of thymine would be equal to adenine, that is 20%. So the rest of the rest would be now guanine plus cytosine. So what will be the percentage of the guanine cytosine? You will have to minus the percentage of adenine and thymine with total percentage of the nitrogenous base, that is 100, and as a result of that you will get that around 60% of the guanine plus cytosine is generally present in DNA. Now, according to the Chargaff's rule, guanine and cytosine residue should be equal, right? So that's why uh, how guanine and cytosine should be equal. If total G plus C is 60%, then you will, in that case, you will have to divide 60 by 2, and as a result of that, you will get that guanine and cytosine is equal to 30%, and here you here you get the answer that in that DNA total 30% of the cytosine present. So this is all about today's class. If you have any problem in understanding anything, you can call me and you or message me. So thanks to all of you. Bye bye.